Class. So let's just pick up where we left off last time, and we're going to talk about DNA replication. So hopefully you know all about nucleic acids and all that good stuff. And now we're going to start working on how DNA replicates. So Watson and Crick came up with their model that Rosalind Franklin helped them with, and from that model they figured out a possible method of replication. So they called that model semi-conservative. And essentially, what that means is that the two strands separate, as you can see here, and then once their strands separate, new strands are built to corresponding nucleotides. So if there's an a T is inserted, G, A, T, Z, and so on. And then the other strand is built in the opposite direction because the two strands are anti-parallel. So these nucleotides are free. They're in the nucleus itself, and there's lots of enzymes involved in separating these strands, keeping the strands apart, adding in primers, which are required because of the shape of the polymerase, and then all that other stuff. The other two models that were proposed by other scientists was called conservative and dispersive. I, there's an experiment by Meselson and Stahl who looked at this, and what they did was they said, okay, if it's conservative, there we go. So we have our two strands of DNA, right? And so these would separate, and then we would have a new strand built on that. I'll just use green. So we'd have a new strand built on that and a new strand built on that. This is what we call semi-conservative. The other possible way would be if we had the two strands came apart, and in the end, the two strands were conserved. So this is fully conserved, and then a completely new strand was being built. And then the last possibility was that this somehow exploded, and we would have fragments of old DNA interspersed with new DNA. Not sure. I don't. Not sure exactly how this would happen, but that's the proposal. So this one here is semi-conservative. This one is fully conservative, and then this one is dispersive. So those are the three possible ways that DNA could replicate itself. So Meselson and Stahl did an experiment. They used heavy nitrogen. That nitrogen is normally 14, but this is 15. It has an extra neutron. And so when they spun this out in a centrifuge in a gel, the lighter in 14 would leave a band, and the heavier in 15 would leave a band that's further down, right? And you probably know this. In centrifuges, heavier things go to the bottom and to the pellet. So if it was fully conservative, you would have one strand with the old, which would be N14, and one strand with the new, which would be N15. So we would expect two bands, and they didn't get that. So they could rule that out immediately. What they got was a single band, and so that meant that these had to be the same weight, right? Which you can see there's equal amounts of red and green on both of the new strands. This could also be the same weight. So they went to the second round. So in the second round of replication, remember green is our new strand. So in our second round of replication, the two strands would separate. So this is our original, I guess. And then so I'm just taking this one strand. So we'll just superimpose it over here. This is the same strand just to give me some room. So that would separate, giving us an old strand and a new strand. So these are just split apart. And the new strand, which is green, would be built on that. So we would have one that's semi-conserved still, but in the second round of replication, we would get one that's fully heavy nitrogen. So this would be heavy nitrogen, and this would be light nitrogen plus heavy nitrogen together mixed. So they did another round of replication to distinguish between these two. Here, this one, if it was still dispersive, even after the second round, they would all be, um, blown up equally and then stitched back together equally. So this would never change. It would always be partially um, light nitrogen and partially heavy nitrogen. Same thing here. So we would expect from this one a single band because they're roughly the same weight. And this one we would expect two bands. One that would be a little lighter because it's half nitrogen 14 and nitrogen 15. So you could say that's nitrogen 14.5 because we're mixing them together. And then you'd get another heavier band that was just straight up nitrogen 15. So in the second round, uh, we would expect to get a single band here, and that's what it's showing, all right, from the dispersive. We didn't see that. And we didn't see this either. We didn't even have to do this experiment because conservative was already done at the first round of replication. The second round of replication, they did see this banding pattern, which meant that the new strands, these right here, right, and the older strands were a mixture of N14 and N15, which is this right here. So that's when scientists knew that Watson and Crick had correctly guessed, even though this is really the only, I guess, novel thought that came out of their paper because all the other information they stole from Maurice Wilkins and Rosalind Franklin, mostly Rosalind Franklin. All right, so now you know how DNA replicates. It's semi-conservative. The two strands separate, and then new strands are built on that. So how does it work? Well, scientists spent a lot of time trying to figure this out. There's a lot of mutations that occurred in different organisms to allow them to do this. Mutations that were actually discovered or introduced, and we're going to talk about how to introduce mutations once we finish transcription, replication, transcription, translation. Okay, so the first thing that has to happen is the DNA has to come apart. So we have the helix, right? I'll just draw T strands. It's not the best helix, but anyway, it has to unwind, right? The strands are held together with hydrogen bonds. Depending on what the nucleotide pairing is, it's either 2 or 3. And so, this is like opening a book. If you want to copy the book, the first thing you have to do is open it. So the first thing that has to happen is that a DNA helicase, yes, that's a protein. It ends in ASE, so it's also an enzyme. Hopefully this is a refresher. DNA helicase simply unwinds it. So imagine if it's a rubber band that's wound tightly. So that rubber band, if you wanted to open it up, you would just simply need to unwind it somewhere in the middle, and then you would have the two pieces of the rubber band separated from one another. So that's a local event. It doesn't happen for all the DNA, so you would just have, like, say this, and you would have an open area, like so, and this would be the area of the helicase. And then this, you would have a 5' end. If we follow this strand, this is 3. If that's 3, then this has to be 5. And if we follow this strand, this is also 3. And then so we can just write it here. If this is 3, this is also 3. And that's 5, this is also 5. And if that's 5 over here, then this is also 3. Right? And remember, 3 has the hydroxyl, That's the only one we can build on. This one has a phosphate. We can't build in that direction. Right? It can only go in one direction. So once the helicase opens it up, this is called a replication bubble. And then these ends are called replication forks. And the forks are different from each other in the way they do replication. It's the difference is really on each of the strands, and I'll show you that. So the helix must untwist, right? Once it's untwisted, there are proteins called single-stranded binding proteins that come here and bind to it. And basically, these are held together by charges, right? Like hydrogen bonds. So you have hydrogen positively charged, oxygen or nitrogen negatively charged. And so they're held together that way, like a magnet. So if you wanted to interfere with the way a magnet like held something on your refrigerator, you could just put like a thicker piece of cardboard on it or something. And then it would be too far apart to actually cause the magnet to stick on your fridge. Same thing going on here. We have the single-stranded binding proteins. They're just interfering with the hydrogen bonding so that the two strands can't go back together. And they're called single-stranded binding proteins because they only bind to, you guessed it, single-stranded DNA. So they're not going to bind. They're not going to bind anywhere here where the strands are double-stranded, only to the open area of DNA. And that's why they're called SSBPs, or single-stranded binding proteins. Now, if we have an open area, if we're looking at the open bubble, and we look at one of the forks, right? So the way that I've drawn this is the 5' end, this is the 3' end, and this is 3' and 5'. So once this is open, the enzyme, which does replication, which is called the DNA polymerase, and specifically this polymerase is numbered 3 because it was the third one discovered. There's a DNA polymerase 1 and a 2 and so on. Roman numerals usually represent polymerases that are found in bacteria. Greek letters like sigma and junk like that are ones that are found in eukaryotes. So it doesn't really matter because prokaryote enzymes can still copy eukaryotic DNA just because of the universality of the genetic code that we talked about earlier. So generally, when we're in a lab and we're trying to copy DNA, we use DNA polymerase 1, but we want to use a thermostable one because we're going to heat up the strands to break them apart. Helicase only opens strands in areas where there's weak hydrogen bonding. So, quick question, do you think that's areas that are AT rich or areas that are GC rich? And the correct response is A and T because it's easier to break 2 hydrogen bonds than it is to break 3 hydrogen bonds. So the origin of replication in organisms generally is what we call AT rich. And that's because it's easier to open. So anywhere this helicase is going to open is going to be called the origin of replication, and that origin of replication is going to be heavy in A's and T's. Simply because it's easier to break 2 bonds than 3. So once this is open, single-stranded binding proteins keep it apart, and the next thing that has to happen is this polymerase has to go on to the DNA. But the problem is that in biology, and hopefully you know this, that shape is critical. So the shape of the polymerase, this polymerase 3, DNA pol 3, it only fits on double-stranded DNA. Well, this is single-stranded, right? So it can't just kind of hang out here or whatever. We need another strand. And the other thing is it can't build. It needs that free 3' hydroxyl group on DNA, right, in order to build. So another enzyme called primase has to come in. Shoot, let me draw the. So this is 5', 3', wait, let me make this 3 prime. It'll be easier to do. 3 prime, and I'll make this 5 prime. And so we'll make this 3 prime and this one 5 prime. So I'm just going to focus on this strand. These are separated strands, right? So this is this same area here where it's separated here and here. Okay, so the primer has to come in and it has to lay down a short piece big enough for the polymerase to sit on. So usually that's somewhere around 10 nucleotides, 10 base pairs, 10 to 20 base pairs. And so this would be synthesized in the 5 prime to 3 prime direction, because remember that hydroxyl group we have to take out water to put another nucleotide in there. Now, what's weird is this thing is made out of RNA and living cells. If we were going to do this in a lab, we would never use RNA, because remember we talked about RNA is really unstable because it's single-stranded, and so it would get broken down quickly by enzymes called RNases and other things. It's not very well protective. Its half-life is in minutes instead of years like DNA. So we would use primers that are made out of DNA instead of RNA, just because they're more stable, they last a lot longer, you wouldn't have to pay for them over and over. But regardless, no matter if it's inside a cell or outside of the cell, you have to have a primer. That primer provides two things. It provides it with double-stranded DNA, so now the polymerase has the right shape to fit on there, and it also provides that free 3' hydroxyl group that's required for the polymerase to do the dehydration reactions to move forward, right? And it's always going to move in the 5 to 3' direction. So new nucleotides are directed into place. They're just kind of floating around here. Let's say this is an A. All right, so the letter that would be inserted next would be a T. That nucleotide would come in, it would get cleaved off. There are enzymes that come behind it and check it for accuracy. We call that DNA proofreading. And then the backbone of the helix has to be fused together to form the new helix. So we'll talk about that as soon as we talk about the difference between the two strands. One is called the leading strand, the other is the lagging strand. So one thing I want to press upon you is this process is super accurate and super fast. Generally about 50 to 500 nucleotides are added every second with an error rate. And so this error rate is after the proofreading and everything else that's going on in the cell, of one mistake per 1 billion nucleotides. So every time your DNA is replicated, you have 3.4 billion nucleotides in your genome. You're going to get about 3 mutations, right? There's nothing you can do about it. It's just, I mean, it's pretty good. Like if you were going to retype a whole book, that would be the equivalent of like a textbook that's, 20, 30 stories high and only make 3 letter mistakes. That's pretty impressive. Anyway, so that's what's going on here. So I'll just kind of get into this in individual slides. So DNA untwist is the origin of replication, like we talked about. Remember, the origin of replication is rich in AT. And we talked about why that is, because there's only 2 hydrogen bonds. Eukaryotic cells have linear chromosomes. We've already talked about that. So they have multiple origins of replication. You would never be able to copy your DNA in any reasonable amount of time without having lots of origins of replication. Prokaryotes usually only have 1, but their DNA is small. We're talking about 4 megabases, whereas ours is about 4 gigabases. 4 billion versus 4 million. So this would take 1,000 times longer to copy than this at equal rates of the polymerase. So we talked about this. The DNA helicase comes in, opens the origin of replication. It only opens here. It can't open anywhere else because it's not strong enough to break a lot of GC bonds. And then we talked about single-stranded binding proteins come along and they bind to hold that replication fork open. So here's the bubble here, and then here's the replication fork here. And this just shows you what it looks like under the electron microscope. So replication proceeds in both directions simultaneously, and then it continues until say this end intersects this end, and then we get two new DNA molecules. So we talked about the bubble. We talked about the replication forks. There's two forks, right? And they're quite different from one another, and we'll talk about that. DNA is added in both directions, so it's bidirectional. All right, nucleotides come in the form of triphosphates. So when we talk about this, we call them DNTPs. This stands for deoxy. And then we add this into a reaction. Like if we're going to copy DNA in a test tube, we would use this. So these are deoxynucleotide. Remember, not nucleoside, nucleotide. And then these are triphosphates. All right. So this is a nucleotide, right? It has the base, the phosphate group, and the sugar. And so this would be what would be incorporated into the growing chain of DNA. It can only add on the 3' end. So this is the 5'. This is the 3'. So this is the only end it could add. And then again, it provides another 3' hydroxyl group here after it's incorporated. This is cleaved off. This is where the energy comes from, just like your ATP energy comes from the same source. And hopefully that's just a review. So that the energy of cleaving these phosphates off to make it a monophosphate and then get incorporated into this dehydration reaction allows the polymerase to move forward. And that's where the energy comes from. Okay, so we talked about this. The polymerase can only start on double-stranded DNA. It's almost like trying to get a regular train, to run on a single track. It would fall over, derail constantly. So the only way to make it stable was to add a second track. I'm not talking about a monorail. The primer is what provides that second track, and it also provides that 3' hydroxyl group that we need. Remember, DNA is anti-parallel, so this is I'm going to have you draw this for part of your problem set. This is the 5' end, this is the 3' end. So if this is 5, this is 3. If this is 3, then this has to be 5, and that's what we mean by anti-parallel. The replication forks are anti-parallel to each other, so I'm just going to draw this out. This one is 5, this one is 3, so this one would be 3, and this one would be 5, right? And so if we were going to prime this, we would prime this, if this is 5, the primer here, we put down here, would be 3 here and 5 here, right? And if we put a primer on the other side, this primer would be 5, and this primer would be 3. Right? Remember, we can only add on the 3' end. So the DNA polymerase that starts replicating this would only be able to add in this direction. Right? This would only be able to go in this direction. Right? The other fork is a little different. So if this is 3, then this is 5. Let's prime it on this side. If this is 3, this is 5, right? And this one's 3. So we could not, on this strand, could not go this way. On the other strand, right, the lower strand, we prime this here, right? If this is 5, then this is 3, and if that's 3, this is 5. So this one could use purple. This one on the lower strand could continue to replicate, right? As opposed to this one on the upper strand. This one could not go this way. So you can see on this side, the upper strand is not able to continuously replicate DNA, only the lower strand. In this end, only the upper strand can continuously replicate DNA and not the lower strand. So it's just opposite depending on which fork you're talking about, the left fork or the right fork. All right. So remember, only add to a free 3' hydroxyl group, and then it can only add if the polymerase is able to sit on a double-stranded piece of DNA. Otherwise, like a train, it would fall off. Okay, so solution to this is add an RNA primer. The enzyme we talked about already is called primase that provides the double-stranded molecule that's required for the polymerase, shown here. Remember, primase. This is the polymerase. This is polymerase 3 because it does the main copying. And so I just want to point that out because we're going to also talk about polymerase 1, which you don't want to have RNA mixed in with your DNA. So the primase sits down the RNA primer here. And once that's done, then the DNA polymerase can start adding here. So this shown here in blue is DNA. This shown here in red is RNA. So you have RNA mixed with your DNA, and that is not good because RNA is unstable, and you don't want your chromosomes to be unstable. So you have to remove that. So there's another polymerase, different shape, and the artist drew it as a different shape here, that can go in and remove the RNA and replace it with DNA. And this is called polymerase. This is polymerase 1, Roman numeral 1. Whereas this is polymerase 3. And remember, in this case, the Roman numerals represent bacterial. Sorry, this is messy. It's a 3, polymerase 3. And then polymerase 1 replaces the RNA primer. Okay, so the leading strand is the strand that can be continuously copied. So remember, this is 5', this is 3' here. Sorry, 3' here. So this is the strand, the upper strand is the one that's continuous. This is only on the left side of the fork. So upper strand is continuous, and you have a problem set like this in your problem set question like this in your problem set 2. So upper strand is continuous. That means once it starts, it can continue all the way until it intersects another replication fork going in the opposite direction or reaches the end of the chromosome. And then the lower strand, in this example, right, and we're only talking about the left-hand fork, this is what we call discontinuous. So the lower strand here, and sometimes this is called the leading strand. Leading strand, and this one is sometimes called lagging. And the reason is that this polymerase can go this direction, right, forever, no problem. This polymerase here can only do this in short pieces, so it has to build a short piece. Let me change colors. Maybe this will be weird. So it has to build a short piece, right? And once it's done that, it has to hop back, build another short piece, hop back, build another short piece, hop back, build another short piece, hop back, and so on and so forth. So because the polymerase has to keep hopping backwards, it can't continuously replicate this lower strand. And the reason is because it can only go from 5' to 3'. It can only replicate this way. This strand can only replicate this way. And you have the exact same problem on the right fork, except the leading strand is on the bottom and the lagging strand is on the top. So anyway, these pieces here are called Okazaki fragments. They were named after Dr. Fragment bad biology joke. Okazaki, and he discovered it because in bacteria that he was studying, those bacteria, they didn't have an enzyme called DNA ligase. DNA ligase is the only enzyme that can link these fragments together. Use a different color. So DNA ligase is required to link each of these fragments to sort of fuse them together, kind of like a welder. So if it's not present, you wouldn't have these fused together like it's shown here. They would just be apart, and then when you heated up the DNA like you would to copy DNA in a test tube, you would see all these little pieces. So that's what Okazaki saw when the bacteria had a mutation in DNA ligase, so it no longer worked. He saw all these little fragments, and he was like, "Oh, I'm going to call those Okazaki fragments." Later on, they figured out that these Okazaki fragments are basically the short pieces of DNA that are synthesized on the lagging strand, a discontinuous strand. Okay, so DNA ligase is required to fuse the gap between the fragments in the DNA backbone back together. And we use that all the time in the lab. If we're trying to insert a piece of DNA, let's say we're trying to put in salmon DNA into strawberry. Let's say this is strawberry. We can take the salmon DNA and we can put it with the strawberry DNA, but without DNA ligase, we couldn't link them together. So we need this. There's also another trick like we talked about the topoisomerases. Isomerase. We're gonna come on. Isomerase. Isomerase. The topoisomerase that we talked before that cut the DNA to prevent supercoiling and prokaryotes. We can use this because it cuts double-stranded DNA and then ligates it back together. So this enzyme has a ligase built into it. So either one of those work if you're trying to splice foreign DNA into another genome. But you need ligase. Okay, so to summarize, replication is performed on both strands at the same time. One is continuous replication, right? In this case, we're looking at the left fork. So the left fork, the upper strand is continuous, the lower strand is discontinuous. And so the priming is done by primase on both the leading strand, the upper strand in this case, and the lagging strand, the lower strand in this case. Elongation is done by DNA polymerase 3. All right, remember we're going to distinguish that. Same thing here. DNA polymerase 3 does the elongation. Remember the primer in living organisms is made out of RNA. We would never do that in the lab. We would always use DNA. Nobody knows why it uses RNA. It's still a mystery. To replace the RNA primer in DNA, remember that's polymerase 1. This one is also polymerase 1. Here you need ligase, but you only need it once, right? You only have to do it if once you set down another primer, whereas here you have to use ligase to fuse every one of these Okazaki fragments together. So you need ligase on the lagging strand for sure. You may or may not need it on the leading strand, depending on where your origin of replication is. All right. I have a better animation to show you. I'm going to pause this, and then I'm going to show you this animation. So you could just go over to Canvas, and that video is here under animations. And if you click on that, and then you can enlarge it. So you could look at that in a I don't know. The resolution kind of goes down when you put it in a big screen, but it's outside of my recording box, so you can't see the whole thing. I can't enlarge the whole thing. So you can watch that again on your own. It's in Canvas. And then there's actually other videos too. I was kind of looking. Um, so the Amoeba Sisters is pretty good on DNA replication. If you guys want to watch that, it just says DNA replication updated. I like Amoeba Sisters. I like Crash Course. Those are kind of outside things that you can watch to help you understand it. They have some pretty good animations. All right, so there's some issues with DNA replication, and one of those major issues is parental DNA. Um, we have the three prime. Obviously, this is five prime. This is five prime. This end is three prime over here. And so we have the two strands separate, which is showing you. So this is the original three prime strand. This one, this is the original five prime strand in dark blue. And here we have the RNA primer has been laid down, and this is the leading strand, right? So it's this three prime end. It can continuously copy all the way down. This one is the lagging strand, right? And so it's copied in pieces. What are those pieces called? You'll know for the test, hopefully, Okazaki fragments. And then ligase has to come in and fix these. So the primase and the DNA polymerase 3, no problem. But once the polymerase 1 removes this primer, right, it cannot, no polymerase can synthesize on this 5' phosphate end. So every time your DNA is replicated, the strand gets shorter and shorter and shorter and shorter. And eventually, you're going to get into a gene that's maybe needed for, say, glycolysis, which is the process of converting, sugars and fats and whatever else into ATP. And you learn that in general bio, hopefully. You are going somewhere else. So you can't fill in that gap. It would be kind of the equivalent of, let's say you had a 10-foot driveway and some joker came and stole a foot off your driveway every day. Eventually, you would have no driveway, right? And it's the same thing here. Once it hits those genes that are needed um, to do, everyday things, we call those housekeeping genes, um, then the cell will start sputtering and eventually die. And so a lot of scientists, you probably read about the CRISPR guy that's doing injections to activate the enzyme that elongates the ends of the chromosomes called telomerase. Um, and this is DNA telomerase can solve this issue. So we're talking about telomerase, and we're so what happens is the ends of your chromosomes shorten every time they replicate. There's nothing that you can do about it. And then remember, telo is end, like telophase, and then meris is part. So a telomere is just the end part of a chromosome. The telomerase is in all of us. It's turned on whenever you are an embryo because your chromosomes would be too short by the time you're born, the number of replications that occur, and then that gene is turned off after you're born. So a lot of people, anti-agers, want to turn this gene back on, and the idea that it would give them an infinite number of cell divisions as opposed to a number of cell divisions. And I know for a fact that I can grow cells that I've harvested, like fibroblast skin cells from, patients or individuals that I'm trying to study. And then if I turn on the telomerase gene, They replicate about 1,000 times as opposed to the 50 times that they would replicate in with telomerase off. So there is some basis for this. The problem is that as a cancer biologist, I also know that almost every cancer that's been studied has telomerase turned back on. That's how cancer cells become immortal. So I have cancer cells in my lab from a boy that had leukemia in the 70s. And as long as I feed those cells and talk nice to them, I don't really have to, they continue to grow. And I can freeze them and thaw them, no problem. So people have tried this. This is a pretty famous case where this CEO, Elizabeth Parrish, she went to Bogota to give herself treatments. So Colombia, to give herself treatments. One was a myostatin inhibitor. So myostatin is something that inhibits muscle growth, and if you knock it out, so statin. If you knock it out, and this is a good example. So this is a myostatin knockout mouse right here. That it's called Mighty Mouse. Someone like had a sense of humor. This mouse did not go to the gym. It wasn't working out. This is just muscle growth due to the inhibition of myostatin. This is a normal mouse. It doesn't. It has myostatin present. So anyway, she did that. Right. She had a myostatin inhibitor tested for muscle loss, and then the other one was a telomerase gene uh, that she claims has reversed her cell's biological age by lengthening part of her genetic material called telomeres. So that's true, right? And so how does that work? So she probably will end up getting cancer from this, but who knows? So anyway, telomerase is an enzyme. It also contains RNA, so it has its own RNA, and this purple part is the protein. And the sorry, I don't know, my drawing pad is weird. Protein, and that protein has enzyme similar to polymerase in it. So what it does is it acts as its own template. So this is the primer, and so it doesn't have to sit down a primer. It allows for the polymerase to have or itself, the polymerase inside of it, to make double-stranded DNA, and so it can go along and add just repetitive sequences. It's just junk DNA. It doesn't code for anything. It's the same as, let's say, we'll go back to the driveway. So if someone's going to take a foot of your driveway, so let's just say you poured a foot of dirt at the end. So they just took that. And then the next day you poured a foot of rocks and they took that. Or you poured a foot of sand and they took that. So in the end, your driveway's still fine uh, because you're just putting a bunch of junk on the end of it that you don't care it's going to be taken or not. And that's the same thing here. So in a normal stretch of DNA, this end would get shorter and shorter and shorter, and eventually your chromosome would look like this. But telomerase just adds stuff on the end, right? Long stretches of just junk. And so when you can't stop the replication process, but every replication cycle, you aren't in danger of getting into a gene that's required for housekeeping. So telomerase just adds on artificial repetitive sequences based on its RNA that just extends the end of the chromosome. So when the primer gets removed, it's not actually cutting into the original chromosome. It's pretty simple, actually. This is a image of the enzyme telomerase. This is called FISH, fluorescent in situ hybridization, and it's lighting up the ends of each of these chromosomes, showing you where the hot spot of telomerase activity is. and you could look it up on a video on YouTube of how this works. It's not that big of a deal. All right, so speaking of telomeres, they can be very complex. Just want to show you this. So we know that the chromosomal packaging, this is a, there you are. Okay. So these are identical copies of each other, so they're sister chromatids. And they're held together by a centromere. Remember, centro is center and meros is part. So telomere is on the end here. That's the end part. And then here's that 20 nanometer chromatin packaging that we talked about when we were talking about histones. So this is the telomere here, and you can see that there's lots of proteins involved in this WRN, TRF1, TRF2 that help protect the end of the chromosome from degrading from nucleases and other things. So the telomeres are quite complex. All right, so we're going to jump back and talk about proofreading. Remember, so the polymerase probably makes an error. Bacterial polymerases make errors like 1 in every 500 nucleotides. So you can get polymerases that are worse or better at incorporating the right nucleotides, but it's nowhere near that 1 in a billion that we talked about. In order to get to that, you have to have lots of enzymes proofreading, looking at the DNA, and fixing those mistakes after the polymerase has gone by. So one of these mistakes that occurs is called thymine dimers. So thymine dimers, you have two thymines, so two T's next to each other in the same strand of DNA. When ultraviolet light hits that, it covalently links them together. So if you remember anything about enterobiology, we talked about ionic bonds and covalent bonds, and by the way, I have a whole 181 course on my YouTube channel. So if you need to refresh this, you can go back and look at other videos from my 181 class. But anyway, so we have hydrogen bonds, super weak. We have ionic bonds, they're weak in the presence of water. We have covalent bonds that are super strong. And covalent bonds are almost always irreversible unless you produce some sort of heat. That humans are allergic to heat, so are other living things. So once a covalent bond is formed, it's easier just to get rid of that piece rather than try to break that bond. And that's what's going on here. So this ultraviolet light has hit this, struck this. These are two T's next to each other. This is a T and this is a T. Same strand, and linked them together, right? So that's a covalent linkage. And so we could break that bond to return it back to normal, and that causes a bulge in the DNA, by the way, which would really screw up the polymerase when it came up here. It wouldn't know what to put in here, so that would cause a lot more mutation. So think about that when you're laying out in the sun. This is what's going on inside your DNA. You're creating thymine dimers. So we have to clip it on each side, right? DNA nuclease is an enzyme. Nuclease is an enzyme that cuts DNA, and we use these all the time in the lab to make cuts in DNA so that we can put new DNA in. There's something called an endonuclease. That means it cuts on the inside. So this example here would be an endonuclease. It's cutting within the DNA. And then there's an exonuclease, and that cuts from the outside or the ends. And in this case, it would be the telomeres of the chromosomes. But it could also be on like RNA or whatever. So when we talk about nucleases, we say endonuclease or exonuclease. It's our friend, the same enzyme, DNA polymerase, in this case, 1, right? Same one that repaired took out the RNA and replaced it with DNA from the primase. It's the same one that repairs, once this has been excised out, repairs that gap. And then again, a polymerase cannot fuse the backbone of the phosphodiester backbone of one strand to another. So that requires DNA ligase, and DNA ligase seals that up, and then we have a newly repaired piece of DNA. So there's a disease called xeroderma pigmentosum, and that disease is also known as vampire's disease. So look it up. And so basically, these people cannot repair thymine dimers, and so they're super susceptible to getting skin cancer. And it's like having a sunburn every time you go out in the sun, right? So you're laying out in the sun and you're accumulating thymine dimers, but your cells are able to repair that to a certain point. So if you lay out in the sun too long, you only have so many workers in your cell and they can only do so much. So like at some point they're going to be like, oh, that's too much. I can't continue fixing this DNA. I quit. And once it can't repair the DNA damage in the cell cycle. There's an enzyme called P53, and that enzyme tells that cell to go kill itself. It's beyond repair. So it undergoes what we call apoptosis or cell suicide, and then your white blood cells come in and gobble those cells up. And that's what causes your skin to be red and your skin to peel and all this stuff. You're actually doing terrible damage to your DNA that's irreparable. So basically, the cell just jumps on the landmine and kills itself. All right. So let's get into a little bit about these polymerases. So bacteria have three main polymerases, the 1, 2, and 3 that we talked about. These are actually discovered in this order. Polymerase 3 is the main player. It's the main polymerase that elongates the DNA. It also has a 3' to 5' exonuclease ability. So remember, it can exonuclease. It can cut out um, nucleotides in a 3' to 5' direction as it's going along. So it can fix its own errors. Remember, it's going to 5 to 3, so it can back up and make small changes in the 3 to 5 direction. So it's mainly adding on the 3' hydroxyl, but it can come back a couple of nucleotides in the 3 to 5 direction and then repair that and then continue in the 5 to 3 as a polymerase. Polymerase 3 or 2, sorry, is fixes DNA damage, right? It has a 3 to 5 exonuclease activity as well, just like polymerase 3. Polymerase 1 is unique because uh, it has the 3 to 5 exonuclease activity, just that we talked about, but it also has a 5 to 3 exonuclease activity, and that allows it to remove the RNA primers. So this is polymerase 1 is the only one that can go forwards or backwards, cutting out DNA nucleotides. And then, like I said, no polymerase can go any direction except 5 to 3 as a polymerase, which means it's adding DNA nucleotides. So those are the three main differences. This can get this can be really important if you're like, let's say you're using a probe. There's probes that we use all the time. This one is called a black hole quencher probe, and so on one end it has a fluorescent dye on it. Let's say it's green. I'll just say it's green. It's green fluorescent protein, so it's gonna emit green, and then on the other side I'll make it black. This is something called a black hole, and you guys know what black holes do. They gobble up light. So all the light from here goes into here. So when you have a detector up here looking at this, it cannot detect this um, light source. As long as these two are close together, the detector can't detect the light. Right? So I don't know. A terrible drawer. Let's just say that this is a camera or something. So no light is going to be emitted there, and so therefore you won't see anything. Now, if you use polymerase 1, it could come in here, and once it hit this, it could start chopping this. So remember, it's going 5 to 3, and then this would be 5 to 3. So any of these polymerases, polymerase 1, polymerase 2, polymerase 3, would be able to cut this up, and that's what it would do, cut up this probe. And once it started getting cut up, this black hole would go off, this would go off, and then you'd get light from the green because the black hole's not next to it, and then you would get a detector. So this is what we use for quantitative PCR, and this allows us to do gene expression analysis. And if we didn't remember the exonucleolytic activity of this, we certainly wouldn't know what polymerase to put into this reaction to figure out where we could look at real time or quantitative PCR gene expression. So you guys need to know it. All right, I'm not going to get crazy about this. Um, Generally, this is not really talked about because we don't use eukaryotic polymerases because generally they're not thermostable. Only bacterial polymerases are. But just so you know, there's alpha, beta, gamma, sigma, epsilon. Um, you know, and so this is synonymous to RNA primase. Uh, Polymerase B is in DNA repair and base excision repair. This replicates mitochondrial DNA, which is mitochondrial DNA is circular like bacteria. And just to remind you, you have mitochondria and they produce ATP in your cells. They also have their own DNA, so that has to be replicated. This has 3 to 5 exonucleolytic activity. It's the main polymerase involved in the lagging strand synthesis. Question mark. And then this one is the main polymerase on the leading strand. So these two work together to replicate DNA. And so if you want to look at how they're connected, there's a really cool video that was made by the Howard Hughes Medical Institute and let's see if I can find it. I'm going to pause this. So what I've done is gone to the DNA Interactive site, um, and basically you can watch the videos here. So if you look at the code, click on the code, that should take you to the pieces of the puzzle and how the code was discovered. You can watch Watson and Crick assemble the code. Um, Copying the code. Okay, yeah, we want to do that. We want to look at copying the code, not transcription though. We want replication. So this is replication. I don't know why that's all funky, but so this is real time. And you can make it bigger. So here's the leading strand, right? This is the two strands getting split apart. This is the leading strand getting replicated in this direction, right? This is helicase going this way, unwinding the DNA. This is the leading strand polymerase. This is the lagging strand polymerase, and they're all kind of linked together. I'm surprised this is the large. I can't zoom in. Anyway, so so this blue thing is the helicase. This is the leading strand. This is the lagging strand. So the lagging strand is kind of looped out. And I'll draw an explanation of what the purpose of this looping is. But this is really how it occurs inside the cell. Okay, and then there's lots of other cool stuff on here. Like you could, let's see, there's not an animation where you can actually be the polymerase. What is this? Oh, before I do that, so there's other videos on here on replication that you guys can go through if it helps you. So all of these, this is on the nucleotide polymerization. This is a leading and lagging strand. So all four of these videos have to do with DNA replication. So if you're not sure or you have any questions about it, then that can help you. All right. So other enzymes that we talked about, and I mentioned this gyrase, also known as topoisomerase. So if you're spinning this thing as fast as a jet engine, 10,000 RPMs, you're gonna create a lot of torsional stress in front of this uh, uh, untwisting of the DNA. Right? You can just take a rubber band and pull it in the middle and see how the ends stress out on it, and that could cause your DNA to break. And if your DNA breaks, that's really bad. So you don't want that to happen. So what topoisomerase does is it can cut each of the strands, and then so imagine that you're cutting one or both of the strands of the rubber band, and then it can spin. It just hangs onto one strand and flips itself around in a circle. So basically, it's unwinding the stress on the front of the DNA. It's hard to do this without an animation, but anyway, it's just clipping. Like if you're clipping one side of the rubber band, it would allow it to spin so that you relieve the stress as you're pulling it open. So get a rubber band, twist it together, pull it in the middle. You'll see that the ends get really tight, and then if you clipped one of those uh, pieces of the rubber band and then unspun it, you would see that you can release that torsional stress in front of it so that the DNA doesn't break. So this enzyme is called gyrase or topoisomerase. We use this in biotech all the time. So this is just an overview of all the stuff in the replication fork. We have the topoisomerases. They're relieving torsional stress. The helicase that's opening it up. We have the RNA primer being laid down by primase. The DNA polymerase is a dimer, like we just showed you in that video. There's a clamp called a beta clamp that holds it in place. And then here's our leading strand, and here's our lagging strand. So this is just a drawing of what we talked about. And then remember, we need ligase to fill in those gaps on the Okazaki fragments. Polymerase 1, remember, its job is to remove the RNA primer and replace it with DNA. All right, so yeah, so that's the end of the chapter. I promised I would tell you guys about how this is. So why we have to loop one of the strands out. So let's say that this is 5 to 3, right? You can't copy this. Uh, so this would be 3, and this would be 5 on the other strand. So you can only copy, make a new copy in this direction, 5 to 3, because the 3' hydroxyl is over there. The other strand is going to be 3 to 5. So it would be 3 to 5, right? But if you loop this around, this is 3, right? And we keep looping it, and then this would be 5. And we keep looping it around. So let me just erase this part. Now we made a loop of it. And so now hopefully you can see that by looping it, now if that's 3, this is 5. And if that's 5, then this is 3. So now we can go this direction. So by looping it, the polymerase can go on in the same direction on both strands. And that's why it has this loop out to make the strand be 5 to 3 instead of 3 to 5. All right. That's the end of the lecture. So we're finished with the lecture 2, DNA replication. If you guys have any questions, let me know. Send me an email. And I will record another lecture tomorrow afternoon.